right, so what's left here to do in your lab report, now that you have your data, okay, is you have to do the analysis. So your analysis looks like this chart. You got your sample code, okay? You got to tell me whether it's ionic or molecular. Which test will tell you that? The conductivity test. So you look at the conductivity test results, ionic, molecular, you tell me, okay? And then you tell me what's the identity. What was A? Was A copper sulfate? Was B water? Okay, whatever it happened to be. All right, and remember that those possible identities are in the materials list, okay, right here. All right, so these are your unknowns. You just have to decide which one of these was A, which one of these was B, and so on and so on, which you should be able to use the stuff from lesson 4 to do. All right, could you also Google it? Yes, you could. I can't stop you from doing that and saying, you know, what are the properties of ammonium nitrate? Yeah, I could do that. Okay, I will tell you this though. All compounds containing ammonium, especially ammonium nitrate, get colder when added to water. Ammonium nitrate is what we use in the commercial cold packs where you pop the water bladder and shake it and it gets cold. The crystals in there are ammonium nitrate crystals. That might help you with one of your identities. Okay? All right, so that's all you have to do for the analysis, okay, is fill in this chart. That's the analysis. Okay, for your conclusion, we said before, what you do is copy and paste your hypothesis and then tell me, can you accept or reject it? Did all of the samples have a unique set of physical properties? Okay, if they did, then you say that and you tell me whether that supports or refutes your hypothesis with a little explaining. Your sources of error, 2 sources of error and how to fix them. Okay, they cannot be human error. Okay, if you say, oh yeah, we contaminated sample B's well with sample A, so your results are bad. Well, and so is your mark then, because if you contaminated something and you knew you contaminated it, you should have started over. Okay, that's human error. That was correctable in the lab and you didn't do it. Okay, something not correctable in the lab would be something like, you know, all the samples were at the front, everybody shared them. Could something have happened there? Maybe you can think of something that might have happened with shared samples that could have been a source of error. I know I picked a whole bunch of the green crystals out of the white crystals next to it. Okay, so stop being so spilly next time. Okay. So 2 sources of error, ways to fix them. They have to be things that you had no control over, okay? Or that you couldn't correct in the lab. This is not due until the Wednesday after the break, so you have lots of time to work on it, and come in and see me if you want me to look it over before then and give you some feedback. I would suggest doing that as this is your first lab, okay? If you want to get a good mark. Otherwise, you're just going to use it for mark feedback, which is not as good. Okay, come in and see me before you submit it, and you'll probably get a better mark. Okay, questions from you guys? Okay, pretty good job today there in the lab. Okay, I didn't have to get after anybody for, you know, radical safety violations. There was a little happy dance going on. Okay, but other than that, okay, nothing too terrible. Okay, so tomorrow, guys, we're going to continue on with reactions. Okay, and we're going to keep working on those.